సంఘలో అప్పుడాలు ఎక్కడ అమ్ముతారు బాగా?
నీకు తెలుసు కదా.
వచ్చాడు మా బాబు. వాయిస్ చెక్ చేయండి ఒకసారి. ఇందులో ఉంటే హలో, వాయిస్ వినిపించట్లేదు. అన్మ్యూట్ చేయండి సార్, అన్మ్యూట్. ఫుల్ వ్యూ ఎలా కనపడుతుంది? వాయిస్ వాయిస్ వినిపించట్లేదు సార్. వాయిస్ చెక్ చేయండి ఒకసారి. అన్మ్యూట్ లోనే ఉంది, వాయిస్ మాత్రం వినిపించదు. స్పీకర్స్ ఉన్నాయా? వాయిస్ రావట్లేదు. నేను మాట్లాడేది వినిపిస్తుంది. నేను మాట్లాడేది పెట్టి నేను మాట్లాడేది వినిపిస్తుందా? జాయిన్ అవ్వు త్వరగా. జాయిన్ అవ్వలేదేంటి సార్? వినిపిస్తుంది, మీ వాయిస్ రావట్లేదు. వాయిస్ రావట్లేదు. రావట్లేదు సార్. నా వాయిస్ వినిపిస్తుందా మీకు? కానీ మీ వాయిస్ రావట్లేదు సార్. మాట్లాడండి సార్. సార్ ఒకసారి డిస్కనెక్ట్ అయ్యి మళ్ళీ కనెక్ట్ అవ్వండి, అయితే రీకనెక్ట్ అవ్వండి. రావలేదు. అన్నయ్య విడిపోతుంది.
సార్ వినపడుతుందా?
ఇప్పుడు వినపడుతుంది సార్.
సార్ వినిపిస్తుంది.
భార్గవ్ గారు వినపడుతుందండి.
సార్ స్క్రీన్ షేరింగ్ ఎనేబుల్ చేయరా?
ఒకసారి చేసాం సార్ ఆల్రెడీ. ఇప్పుడు చేయండి సార్ మీరు.
సార్ స్టార్ట్ చేస్తున్నా.
Good morning, one and all. Welcome to the 1 week EFTP and exploring new innovations in novel drug delivery systems. Today's guest is Dr. V. Bhushan, sir. Principal, sir, Dr. Bhaskar Raju will present the meeting today. Please, Bhaskar, sir.
So, very good morning to all the participants and delegates. I am Dr. V. Bhaskar Raju, Principal and Professor of Srivastava Institute of Pharmaceutical Sciences. So we are very delightful to organize 1 week FDP on new innovations in novel drug delivery systems. So welcome to day 3 of our FDP. And today's resource person is Dr. P. Bhushan Rao, and he is the Principal and Professor of A.M. Reddy College of Pharmacy, Guntur. And on behalf of our management, organizing secretary and organizing committee, I welcome our today's research person to share his valuable insights in novel drug delivery systems. Now I request our faculty, assistant professor P. Shravanti will give a brief introduction about our resource person, Dr. P. Bhushan Rao.
Yeah, thank you, sir. Good morning, delegates. Thank you for registering for FDP. I am glad to introduce our distinguished speaker, Sri Dr. P. Bhushan Rao, sir, who is working as a professor and principal at A.M. Reddy Memorial College of Pharmacy, Guntur. He completed his B Pharmacy in the year 2006 from KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada, and secured 89th rank in GATE. And he completed his master's in 2008 from KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada. He has 15 years of teaching experience, and he filed 3 patents in India, Germany, and South Africa, and has 9 publications. He published 1 textbook on research methodology. And the research grants he received till date are AICTE RPS worth 15 lakhs in the year 2019 and AICTE Spices worth 1 lakh in the year 2019 and Unnat Bharat Abhiyan worth 50,000 in the year 2002. We are very happy to have you here today. I request Sir to speak his views on novel drug delivery systems. Sir, please proceed.
Thank you, Shravanti, for your introduction. And today I would like to give a brief note regarding transprothams. Anyhow, welcome all for 1 week FDP on exploring new innovations in novel drug delivery systems, organized by Srivastava Institute of Pharmaceutical Sciences. Thank you for inviting me. So today I would like to give a brief things regarding one of the novel drug delivery system, that is transferosomes. So these transferosomes, we are intended to uh, use these transferosomes. The route of administration is by transdermal route. So there are many vesicular systems that are present in novel drug delivery systems, but out of which these transferosomes um, uh, now they are proved better when compared to the liposomes, neosomes, and erythrocytes and all those things. So that's the reason why I have taken this particular topic. So, so from many decades, scientists are working on different types of systems and different routes of administration. And when compared to oral route and other routes, they found that the drug delivery through skin would be one of the advantages, because the side effects that we come across from oral drug delivery systems can be minimized. Especially first pass metabolism can be bypassed when we go with the skin as a route of administration. And there is a larger surface area of skin that is being available. So that's the reason, another reason why scientists are choosing this particular route as one of the method for the transferring of various drug delivery systems. So the only drawback with the skin as a route of administration is that is cutaneous and transcutaneous drug delivery. That means that the drug is the permeation characteristics of the stratum corneum that restricts the drug transport. So that is the main problem. If that particular problem is overcome, then automatically we can use skin as the route of administration. So stratum corneum is a superficial layer of the skin, so which is being comprised of keratinized flattened residues of dividing epidermal cells, impermeable to water, and behaves as a tough flexible membrane. So stratum, from this point, which we can understand is stratum corneum doesn't allow the drug substance which are aqueous soluble. So that is the one of the constraint which we face when we use skin as a route of administration. So many technologies we have tried. There are many scientists who have tried many technologies, and they have also worked on liposomes and microemulsions and all those things. But they found that there is an issue with the drug transfer through the stratum corneum. So then they have started exploring on the transferosomes. So actually, transferosome, సార్ డిస్టర్బెన్స్ వస్తుంది సార్ నాకు అక్కడ వెనక ఎవరో మాట్లాడుతున్నారు భాస్కర్ గారు. సార్ భాస్కర్ గారు, సార్ సమ్ మ్యూట్ చేయమనరా సార్, ఎవరో డిస్టర్బెన్స్ వస్తుంది.
ఎస్ సార్ చెప్తాను.
సో transferosome, the name transferosome itself says it is a carrier. So carrying a particular drug substance is the main title, that is transferosome. So in this particular transferosomes, we use yes. So transferosomes are vesicular carrier structures that are specifically laid down to have at least one internal aqueous compartment which is surrounded by a lipid bilayer. So beside an edge activator. So when we think of a composition of a transferosome, the main advantage of transferosome, it is a bilayer where you can accommodate a large amount of lipoidal drug substance. That is the major advantage. And the edge activator that we incorporate. So edge activators are nothing but the surfactants which make the system compatible. That means, so even if there is an aqueous environment and lipoidal environment, so this edge activators helps to make the transferosome stable. And one more advantage of these edge activators are they help in increasing the entrapment efficiency of the drug substance. So from this particular definition, we can clearly understand that. So if the drug substances are when they are having high lipoidal in nature and even low lipoidal in nature, they can be accommodated using transferosomes. So that is the advantage of transferosome. So keeping that in consideration, we are going further for formulation and all those things. So another advantage of transferosomes, they are ultra flexible. So these ultra flexible can be attained because since we have used edge activators, the ultra flexibility can be achieved. So the edge activators used facilitates the solubility of hydrophobic drugs, thereby further enhancing the entrapment efficiency of the drug. So that's a point which I have discussed earlier. So even another very important is the particle size. The particle size that can range from 600 nanometers to 70 nanometers. So the only thing is, even if the particle size is higher, even if the particle size is lower, so the transferosomes can be traveled through the membranes of the skin. But the only constraint is the amount of the drug substance, that is the lipoidal drug substance, that we consider should be important. And the particle size should not be, if it is greater than 600 nanometers, but see that it has maintained some potential and all those things. So these things should be maintained. So when we are going for the formulation of the transferosomes. So this is the structure of the transferosome, where it is clearly understood that there are two layers of phospholipids, where where the drug substance, if it is lipoidal in nature, it can be completely entrapped within those. And in center, we can see that there is an hydrophilic drug substance, where if the drug substance is aqueous in nature, those can be accommodated over there. And finally, there is an edge activator, so which is responsible. It is a surfactant which keeps in bind all the drug substances together. So this is the basic structure of a transferosome. And also, in order to make these edge activator and phospholipid compatible, we use organic solvents, so that they also enhance the what you call compatibility of the transferosomes, or else the formation of the transferosomes can be enhanced. So there is a lot role of the organic layers that are being played in the formulation of transferosomes. So that I will discuss when I am discussing methods of preparation. So from this particular structure, it is clearly evident that these transferosomes can accommodate lipoidal substances. At least 90% of the most of the drug substances which are lipoidal can be made in the form of transferosomes. So that is the advantage. So this is another a pictographical representation how the transferosomes permeate through the skin layers through stratum corneum. So the edge activator that is being present in the formulation of transferosome helps also in the phenomenon of deformation. So the edge activator that we use, so like surfactants and all those, so they help the phenomenon of deformation, which is clearly evident in the pictographical representation that so in the center, when it is traveling, so the deformation can be observed. So the deformation is produced due to the difference in osmotic pressure that is being generated. So that is the point which we have to understand. So the edge activators are responsible. So that difference in osmotic pressure automatically concises or deforms the transferosome structure, and it allows the transferosome to travel through the skin membranes. So that is another important point which you have to understand. So what are the advantages? Some of the advantages of transferosomes. So transferosomes improves bioavailability. So this is one important point which you have to. So those drug substances having low bioavailability, automatically the bioavailability can be enhanced. Bioavailability can be enhanced when we make in the form of a transferosome. So once there is increase in bioavailability, automatically there will be increase in patient compliance. So another thing is high entrapment efficiency. So which means that the range of drug substances, can be, if the drug substance is low lipoidal in nature, and even if it is having high lipoidal in nature, both type of drugs can be accommodated in the transferosome. That is the advantage or the important in case of transferosome. So once if it is can accommodate even low lipophilicity and high hydrophilicity, which means that the HLB range can be, it has a larger scope of HLB range. So that's the reason why the edge activator that we use, it has a larger range of HLB scale. Okay. And next, it releases the drug slowly and steadily and act as a depot. So once the drug, it reaches to the systemic circulation, slowly it forms as a depot. And whenever there is a need, and when there is an unsync condition or sync conditions that is maintained, automatically the drug slowly enters into the systemic circulation. So transferosomes are used for topical as well as systemic delivery of drugs. So the main intention of these transferosomes is to travel or to use skin as a route of administration. So that is the main purpose. So since they have high entrapment efficiency, and since they're having a capacity to deform themselves when they travel through the stratum corneum, so these transferosomes are more advantageous. And larger scope of drugs like proteins and peptides and hormonal preparations, they can also be made in the form of transferosomes. And much research is going on these transferosomes. So many creams and gels are being made. So for example, the transferasomes are incorporated into a gel, and drugs like drugs that are used for treatment of impetigo, NSIDs, and all skin infections. So nowadays, these type of things are being made using transferasomes. So disadvantage. So the main disadvantage is the purity of phospholipids that we get. So most of the times it is a major drawback in the formulation of transferasomes, that the phospholipids that are being available are not pure. So that's the reason. So there is alternative also to make pure transferasomes, but to the extent of purity is not the extent of pure transferasomes or phospholipids are not available. So that's one of the major disadvantages that come across. And the since we use these transferasomes into the skin layers, so the skin structure is also very important for the carriage of these transferasomes into the stratum corneum. So, and that would be one of the disadvantage. And next, hypersensitivity. Since we use these transferasomes very frequently, so that there may be a chance for skin irritation to occur. So that is one of the disadvantages which we need to discuss. So what are the factors affecting permeation into the skin? So as I told you that the one of the major drawback is the skin age and condition of age. For example, when the person age passes from year by year, automatically the hydration process of the skin, or automatically the lipid content that is present to the person automatically declines. And because of that phenomena, the permeation of the drug or the permeation of the transferasomes into the skin layers also decreases. So that is one of the factor which we have to understand, that is biological factor. So the skin condition, if the skin condition is good, automatically the permeation will be good. And if the skin condition is not good, the permeation will be decreased. And metabolism of the skin. So we know that the phenomenon of metabolism also happens on the within the membranes of the skin. So once there may be a chance that the permeation of the drug and metabolism, there may be some interactions which do not allow the easy permeation of the drug through the stratum corneum. So that would be one of the factor which we have to understand. And the site of administration. So once if there is a thick site or rigid membranes, the transferasomes cannot permeate through those. So that would be one of the drawback that we have to understand. And physicochemical properties, drug concentration. So the flux is the parameter that we calculate the concentration of the drug. So the flux is nothing but the amount of the drug substance that is being permeated. So the greater the flux value, the greater is the drug concentration. So the membrane that is being available. So if the membrane permits the drug to permeate, automatically it will have high flux value. And if the membrane does not allow the drug to permeate, automatically the flux value will be declined. So the flux is the parameter that clearly makes us understand to what extent the drug is being available. Next, the hydration of the skin. So hydration, as I told you previously, that so the one more important factor that keeps our skin very flexible is hydration. So if the content of water, it is not good in the skin, if the skin doesn't maintain high hydration value, automatic the permeation of the drug will be decreased. So that's the reason why during the formulation of transdermal patches, we use little amount of humectants. So the usage of humectants helps the drug substance to permeate in a better way. And the temperature and pH. So these are the very important factors which we discuss not only in the transdermal route or transferasomes or whatever it may be, also in the oral drug delivery systems. And that the ionization and anionization. So these are all dependent pH partition hypothesis, which clearly explains regarding the ionization and anionization. So the temperature and pH are the governing factors which affect the ionization and anionization. So it is very important that the unionized form of the drug, those only determines the concentration of that particular drug substance. So if it is in the ionized form, it is of no use where we cannot understand the concentration. So whenever we are formulating, see that the pH that is being maintained is neutral in nature, so that the drug clearly permeates. Okay. Another very important is partition coefficient. So once we are formulating any dosage form, the pre-formulation process, in the pre-formulation process itself, we need to determine the partition coefficient of the drug. So the so the lipophilic content, when it is high amount of lipophilic content, automatically the permeability of the drug substance into the skin layers will be high. If there is low lipophilic content, the permeation will be decreased. So which means that the log K value should be greater than 3. So once if the log K value is greater than 3, that means that the permeation of that particular drug substance will be high. So next we come to in composition of transferosomes. So the transferosomes composition consists of the main two important things are phospholipids and edge activators. So as I told you previously that the transferosome structure itself consists of a phospholipid bilayer. Okay. So the composition of transferosomes, the major part will be occupied by the phospholipids, and the next important part that is occupied will be edge activator. So these two, this is another very important point which we have to highlight is in the formulation of transferosomes, the number of excipients that are being used are very less in number. Hence, there will be less number of interactions and more percentage of stability. So this is the point which we have to understand. So the phospholipids that we take, so they have to maintain the pH of at least 6.5 to 7. That is one important point. So most probably we use phosphatidylcholine and soya phosphatidylcholine and egg phosphatidylcholine. These are the most widely used phospholipids in the formulation of transferosomes. And edge activators. So edge activators, these are, we call them as surfactants, and these are the agents which keep in bind both the aqueous phase as well as the oil phase. And the most widely used edge activators are tweens and spans, and these are being added in order to make the transferosome very stable. So I told you the importance of the edge activators also. They impart the flexibility to the transferosome, and they are also responsible for what you call high amount of entrapment efficiency. So this is also another important thing which we have to understand. Next. So principle of skin permeation. So this is the principle how the drug or the transferosome permeates through the skin layers. So transferosomes cross the skin barrier by passive diffusion. So here the point that we have to understand is the fixed law of diffusion is being considered, fixed law of diffusion. So which means that the solute transfers from one compartment to the other compartment over the course of time, separated by a thin membrane. So thin membrane per unit surface area. So the rate of transfer, that is dq by dt, is directly proportionate to the concentration gradient across the membrane. So the point which I told you, the flux. So this is completely related to this particular parameter. So if the drug substance is the drug substance bypasses or penetrates through the membrane, and if the availability of the drug substance is high, automatically the flux value will be high. And if the permeation phenomenon doesn't happen properly, automatically the flux value will be less. So basing upon the flux value, so what is the amount of the drug that is being available for systemic circulation? So this is the important point, or the principal part which we have to remember whenever we are discussing not only these transferosomes, whenever we are making any dosage form that is traveling through the skin as a layer. So we have to understand the skin permeation is one of the factor. Next, so in vitro testing. So we use Franz diffusion cell. So this Franz diffusion cell consists of a donor compartment as well as a receptor compartment. And in between donor and receptor compartment, there will be a membrane, maybe a skin membrane that may be, it depends upon the nature of the dosage form. We can use pig membranes or dog membranes, or also we can use cellophane membranes. So for case of in vitro, but anyhow, so whatever the drug substance or the transferosomes that we have prepared, we put it in donor compartment, and the skin membrane, we put it in between the donor and receptor compartment. And there will be a receptor compartment where we can collect the sample, and you'll have a stirring bar where the sample will be stirred, so that we can collect the samples from the receptor compartment. And basing upon that, we can also study how much amount of the drug that is being traveled from the skin membrane from donor compartment to the receptor compartment. So this these apparatus, we call it as Franz diffusion cell. So there are various methods of preparation of transferosomes. So one very important method which most widely used is rotary film evaporation method. So this is also called as modified handshaking method. So uh, first of all, before we start the experiment, we take the mixture consisting of both phospholipids and surfactants. So as I told you in the components of transforosomes, they consist of phospholipids as well as surfactants, and we add the drug substance into the mixture of phospholipid and surfactant. And to that, we add a little amount of organic solvent and we go for vertexing. So once we do vertexing, automatically all the components, that is phospholipid, surfactant, and lipophilic drug substance, they will be dissolved in a proper manner. Okay. So next, once the vertex is done, we transfer the solution to a flask for rotary evaporation, where it is rotated at a constant temperature under vacuum to obtain a thin lipidic film composed of active ingredient, adjectivator. So it is the important thing that, so once it is being vertexed, so we use we transfer that into a rotary flask, and where the phenomenon of evaporation is done. Okay. So once it is done, we add a little amount of aqueous medium, okay, in order to hydrate that film form, which swells and forms vesicles, which are bilayer after hydration. If the drug is hydrophilic, it can also be added to the aqueous medium. Sonication or extrusion techniques can be used for reducing the size of the formed vesicles. So it is the prime important that we need to maintain the particle size. So for that reason, we go for the phenomenon of reducing size. So for that, we go for sonication. So once we do that sonication, automatically we can get the transforosomes that are being formed. So this method is particularly useful in delivering the drugs to specific target sites due to the ability of the vesicles to permeate the skin stratum corneum and enter the systemic circulation. So since we have maintained the particle size, so to the desired, then automatically we expect that these transforosomes, once these transforosomes that are being formed, we incorporate into suitable gel or creams, whatever that we require as carriers, and we allow that particular drug substance, allow those transforosomes, and we apply on the surface of the skin. So this is one of the major method, and that is rotary film evaporation method. So the next important is reverse phase evaporation method. So the name itself says that it is reverse phase. So in this technique, phospholipids are dissolved in an organic solvent. So which means that that the phospholipids are compatible, and we dissolve in that organic solvents. So those organic solvents may be chloroform or methanol or ethanol, and we place those in a in a flask. So hydrophilic media consisting of a surfactant such as adjectivator. So we call that surfactant as an adjectivator, and is added to the flask while pouring with nitrogen gas. So depending on the solubility characteristics, the drug is incorporated either in the hydrophilic or the lipophilic media. The resulting mixture is subjected to sonication till it becomes a clear dispersion, so which is homogeneous in nature and is observed minimum for 30 minutes after sonication to ensure that there is no separation occurs. So the name, as I told you, that reverse phase. So we see that we oppositely we add this particular drug substance in water, and we get the transforosomes. And here also we perform the sonication so that we get some desired particle size. So this is another method which is most widely used. The first preferred method is rotary film evaporation method. The other one is reverse phase. So in this case, the main important thing is when the drug is being soluble in water, and in that case, we go with this reverse phase evaporation method. So next one is ethanol injection method. So ethanol injection method, in this, first we dissolve the lipoidal phospholipid substances in organic solvent, and we keep that aside. So in a injection where the particle size can be monitored or the particle size can be maintained. And in beaker consisting of drug, okay, aqueous solution in which the drug is being dissolved, and we put it on the what you call stirrers, and we maintain certain RPM, and we slowly start adding the lipoidal substance that is present in an injection, and we add drop by drop, and we leave that particular mixture for a period of time so that the organic solvent gets evaporated. So once this process is done, during the evaporation process, the organic layer slowly evaporates, leaving behind or leaving the lipoidal layer on the surface of the drug substance. So this method is also most widely used. So this is internal injection, and this is simple one where we can maintain the particle size. So the next very important is freeze thaw method. So freeze thaw method, the name itself says we are freezing and increasing the temperature. So we are making both high temperatures and low temperatures we are maintaining. So that we call it as freeze thaw method. So to obtain the transferosomal formulation, we use alternate cycles of freezing and heating. So both the things we do it. So the multilamellar vesicles that are exposed, that may range from at least minus 30 degrees. So we maintain low temperature of minus 30 degrees so that the freezing phenomenon happens. So once the freezing is done, automatically we raise the temperature. There results in the formation of what you call transferosomes. So this is the method where it is also used, not much but used. So vertex sonication method. So this is one more method which is used. So in this vertexing method, it involves mixing of phospholipids, drug, and edge activator in a base. Base may be a phosphate buffer or whatever the base it is being required. So basing upon mostly we use buffers which are pH 7.4 and all those things, phosphate buffers. So followed by vertexing of the mixture until a suspension. So we stir it. So we use the phenomenon of vertexing where all the phospholipids and drug and edge activator are completely soluble. And once they are soluble, that particular suspension will be milky in nature. The product is then subjected for sonification. So the particular once the formed is, we sonicate for at least 15 to 20 minutes and followed by extrusion. So this extrusion is done. In this extrusion phenomenon, we follow one particular thing that, so we maintain one particular size. In this particular slide, I have shown that 0.2 micrometers. So we pass this particular mixture or this milky substance through the membrane extrusion, maintaining a particle size of 0.2 micrometers. So once we extrusion through a membrane filter it is made up of polycarbonate and so that the size is being maintained. So this is the another process where the transferosomes are being prepared. Next, so once the transferosomes are prepared, it is very important that we need to evaluate. So it is especially in these novel drug delivery systems, lipoidal systems especially, for example, liposomes, neosomes, and all those things. We see that there is formation of the structure, but it is completely not true that the structure that is formed consists of the drug substance. So it is very important that we need to evaluate those parameters. The first very important is entrapment efficiency. So entrapment efficiency is being calculated. Percentage entrapment efficiency is being calculated by using the formula amount of the drug entrapped by total amount of the drug added. So suppose, for example, if the amount of the drug that is added is suppose 100 mg and amount of entrapped drug is 70 mg. So the entrapment percentage is 70%. So the entrapment efficiency depends upon the edge activators that we use. That is one important point. So basing upon the percentage of edge activators, the amount of entrapment efficiency also varies. And the excess of edge activator also creates some hydrophobicity. So that is also another point which we have to understand. Okay. So next, zeta potential. So in order to make a stable preparation, so there should be a potential that exists. So so since we are making the lower particle size, nanometer size, there will be a charge that is being developed. And in order to make a stable transferosomal preparation, we need that there is some potential that exists. So we need to calculate the potential and also the particle size. Suppose to what extent the particle size, we use Jetta as an analyzer or Jetta sizer for this phenomenon to study. And vesicle morphology. So the formation of transferosome can be clearly the structure of the transferosome can be clearly understood when we study scanning electron microscopy SEM. So, and TEM, transmission electron microscopy, where we can understand the size of the particle and the morphology, how the structure of transferosome it is being prepared. So whether it is spherical or not, or whether to what extent the size of the particles that are being formed, and the drug content. So drug content, this is what we do. All the process which whatever we do in nanospheres and all those things, that the same we adopt here also. We make the drugs transferosomes to determine the drug content. We use a UV method where the drug is transferosomes are made to release the drug substance in a suitable solvent, and we analyze that suitable solvent by basing upon its wavelength, and from that we can determine the amount of the drug that is being present. And next, total number of vesicles present per cubic mm. The total number of vesicles present per cubic is determined by using a hemocytometer and optical microscope. So by using the optical microscope and by taking the sample and introducing in an optical microscope, you can clearly know the number of vesicles that are being present per cubic mm. And next, in vitro drug release. So as I told you that Franz diffusion cell we use for determination of in vitro drug release, where the sample that we have prepared or transferosomes that we have prepared, we keep in the donor compartment, and from the receptor compartment we collect the samples, and by using UV spectrophotometer and in particular wavelength, we can calculate the amount of the drug that is being released. So these are some of the tests that we perform, evaluation test for transferosome, and there are certain applications of transferosomes. So transferosomes, the main important is the intention of preparing a transferosome is to achieve a control release. So as I told you in the application, that these transferosomes they form as a depot, and from that depot the drug is being released, and that's the reason why it is being in the applications we call it as control release of the drug. And whenever there is a peripheral drug targeting, for example, diseases like impetigo. So impetigo is a disease where we can see that it is a surface. The disease is caused by just the surface skin irritations and all those things. So these type of things can be clearly, these type of diseases can be clearly treated by using transferosomes. Next, transport of large molecular weight compounds. Since these transferosomes are made by using bilayers, phospholipid bilayers, so it is evident that a larger molecular weight, protein molecular weight, and fatty molecular weight substances can be accommodated. So that is one of the application. And delivery of hormones, that also will get done, and delivery of anesthetics, and all these things can be done by using transferosomes. And I conclude this. So transferosomes display deformable properties that improve. As I told you, they when we make the drug substance incorporated in the transferosome, they improve the bioavailability. So once the bioavailability is improved, automatically the amount of drug substance that is available will be improvised. And once the formation of transferosomes are stable, which means that we can achieve stable type of formulations. And since we have used edge activators and all those things, they enhance the phenomenon of penetration. Okay, so and also they are also responsible for good elasticity property. So that is one of the important use or application of these transferosomes. And the primary factor for the transfer of transferosomes into the inner epidermal layers is the osmotic gradient. So because of the maintenance of osmotic gradient, that is the reason why the transferosomes can be deformed from the stratum corneum and reach to the systemic circulation. So the flexibility can be attained, especially when we make in the form of transferosomes. As we told you in the first slide, that the major rate limiting step for the permeation of these vesicles is stratum corneum. So since these transferosomes are capable of bypassing those or passing through this stratum corneum, hence I call these transferosomes as the superior when compared to the other vesicular systems. So researchers have found that these delivery systems are one of the successful ways to deliver wide range of drugs. So as previously I told you that since it is bilayered, it can accommodate larger molecular weight substances and higher lipoidal contents. So that is one of the important things. Furthermore, it has a promising future as a nano carriage system integrated with the computational method saves the time and effort to get the best possible formulation composition. So since the number of excipients that we are using are only limited, the chance of formation of transposomes are very easy. Okay, so the success rate will be high, and the scale up technique can be also done since the number of the rate limiting factors or the troubleshooting issues are very low. So that's the reason why transposomes can be made to the larger in scale. So there are many advantages. So one other very disadvantage is the cost. So transfers, all these novel drug delivery systems which we discussed, all the cost effectiveness is not is one of the major disadvantage in all liposomes and all those things. And here in this case also, and scale up process, as of now, the scale up process of transposomes are not being available. But so in the lab level, we are getting the formation of transposomes, and so one of the another disadvantage is leaching effect. So which this problem we come across on all the vascular systems. So additionally, limited clinical trials. Yes, I told you that there are a limited number of work still going on with these liposomes. So there is a need, urgent need to conduct more clinical trials to provide safety and efficacy profile for transposome. And if we go on working on these transposomes, so automatically we can improve the scale up techniques also, and so that that will be helpful for the real need of the real need, and many drugs can be incorporated, and transposomes can be available in the market. So these are some of the references which I have selected. Thank you, sir. Thank you very much for giving me this opportunity. And especially I thank Chief Patron, Sri Subbarao Garu, and Program Director, Principal Dr. V. Bhaskar Raj Garu, and Organizing Secretary, Dr. P. Narayan Raj Garu. Thank you, sir. Thank you very much for spending a valuable time and accepting. Thank you very much, sir.
Thank you, sir. Thank you very much for time spending with us at the event. Thank you for the lecture one. Now it's time to question anyone, delegates, any questions? Any questions? Shanti is asking hydrophilic and lipophilic drugs can carry by transposomes. Then why lipophilic drug delivery become issue?
Actually, majority will be lipophilic, sir.
Okay.
Majority will be lipophilic, and minority will be hydrophilic, sir. And in this case, especially when compared to other vesicular system, transposomes are more advantageous because they are bilayered, and they can accommodate larger content also. And basing upon the nature of the particular drug substance and its partition coefficient, we can make it in the form of a transposome, sir.
Okay.
So we have
Ranjani is
also
asking.
made we have also made transposomes for impetigo. We have made three types of transposomes to treat impetigo. So we are successful in the lab scale where for incorporating into the gel and all those things we have done. So we have used the basic one thin film evaporation technique only for the formulation of transposomes. So it was easy, and we were successful with that.
Sir, Anjali is asking. Yeah. Anjali is asking, is there any difference between liposomes and transposomes?
Major difference is the concentration of edge activator and the amount of what you call phospholipid. But since it is a bilayer, so it will be more advantageous when compared to the liposomes, sir.
Any questions? Anyone?
We have made fusidic acid transposomes we have made previously, and we have incorporated into gel, and we published also a paper with those fusidic acid also.
Okay. Person Ali Khan, sir, any marketed products are available? I'm asking.
No, sir. As of now,
Ashok
there
Ali.
are no marketed products, but they are very soon are coming. Transfrosomes, they are coming. Why?
Is
Because
there any
I
company,
told you.
sir? Is there any company manufacturing transfers
Yes,
who
sir.
are on the
Now,
trials?
yes, sir. Sun Pharma is working on that, sir. Actually.
Okay.
Yes, sir.
Which drug, sir? It is in which drug?
Fusidic acid, sir. Fusidic acid.
Fusidic acid. Okay, okay, okay. Any more questions?
Fusidic acid and retapomylin. Retapomylin is also one of the other drug also, sir. These two are they are working actually.
So, more questions will wind up. Questions. Sir, someone is asking Srinivasa, Gopala Srinivasa. Sir, what are the equipment used for size reduction of the transfers?
Homogenizer we have used it, sir. Homogenizer one thing we have used, and we have also used some homogenizer only we have used.
Any more questions? So we'll wind up this session. Thank you very much, Bhargav sir. Once again, for your valuable time. Your busy schedule was spending a lot. Thank you very much for your valuable lecture on transfers. Thank you, sir.
Thank you, sir, Dr. Bhargav Garu, for sharing your experiences and knowledge regarding the transfrosomes. And on behalf of our management and organizing committee and faculty, I thank our resource person, Dr. P. Bhargav Bhushan Rao Garu, for his valuable speech. Thank you, sir.
Thank you, Bhaskar sir. Narendra sir. Thank you, sir.
Thank you, sir. Thank you very much.